Hi, this is Miss Litton, and this is 7th period support for Honors Bio. Say hi.
Hi.
Specifically Chapter 8, where we're focusing on photosynthesis. So let's hit all the key target points. First of all, plants are called autotrophs because they can take inorganic molecules and form organic molecules with them. We, however, are called heterotrophs. So starting macroscopically, looking at a plant, okay, we know they have green parts to them. Those green parts are probably made green by the pigment what? Chlorophyll. And you see those in the leaves. And so we'll look at a leaf in just a minute. We can see the stem. Might be a barky, woody, or a stem that's not. We can see the roots. And from the roots is where the plant will acquire what? Water. And also minerals. But that's where the water comes in. We know that's one of the reactants of photosynthesis. What's another reactant? Carbon dioxide. How does carbon dioxide get into a plant? Through the leaves. What part of the leaf? Stomata. The stoma, right? And that's how it gets into the leaf. What's another reactant we need for photosynthesis? We need energy. Now, it could be a light bulb, okay? Or it could be more than likely the sun, right? You need some form of energy. What do we need the energy for? Why do we need the sun? What does the sun do? It excites what? Electrons. Okay, so that's why we need the sun's energy, to take electrons to a higher energy level. So we need electrons. Where do the electrons come from? Water. So that's why we need water. Why do we need CO2? To build the sugar, right? You're tired. What's the formula for sugar? Oh, stop. C6. Okay, that's carbon. So we need a source of carbon, and CO2 is the source of carbon. Okay? Don't forget the big picture, right? When you're studying all the microscopic pictures, remember the big picture because you're like, I don't know why. It's a mystery. Okay, so now let's go in a little bit deeper and let's focus in on one of those leaves. And when we look at a leaf, as we're zooming in here, when we look at the cross section of the leaf, we can see the stomata are on the underside of the leaf. What would that do? Why would that be helpful? Why don't you want them on the top? I didn't even talk about this in class. You should be able to think about this. Here comes the sun traveling in waves. If the stomata are on the top, what's it going to do to the water right inside that leaf? Make it evaporate. You're going to lose it, and those stomata are going to have to close more often. And we know we don't like closed stomata because what happens? You run out of what? Carbon dioxide, and you have all the oxygen, and then you start doing what? Photorespiration, which isn't photosynthesis, and it's also not respiration. So that's why stomata would be on the underside, so you don't lose as much water that way, right? Okay, when we look inside the leaf, we can see all the leaf cells. This is a standard C3 plant where you have palisade mesophyll here, spongy mesophyll there. Here's those bundle sheath cells that are in blue here. It's not like they would be in blue in a plant. They're just kind of differentiating. If it's not green, you know it's not doing what? It's not doing any photosynthesis, right? But we know exceptions to that too, right? Remember the Krantz anatomy with the C4 plant where it looked like a wreath? And that's because what they were doing is they were doing all of their dark reactions right here and surrounding them with mesophyll cells to protect RUBP from binding to what? Oxygen. And where did you see that kind of setup? In the tropics. There's a lot of sunshine. And where it's going to get hot and where you're going to be doing a lot of photosynthesis and there's going to be a lot of oxygen there, and that's when you have to worry about it, right? All of those types of factors. So that's one way. These little green dots in here are what? The green dots right there? Chloroplasts. The big blue things are what? Vacuoles. Good. So when we blow up one of those chloroplasts, we see this inside. So inside of a chloroplast, which is double membraned, which makes me think of the endosymbiotic hypothesis, right? Remember that? Okay, so now I'm looking at these stacks of green pancakes. They're hollow pancakes, very light and floaty. And those are made out of thylakoid membranes. Say that word, thylakoid membranes. And what happens on the thylakoid membranes are all the na-na-na-na-nas. What's the na-na-na-na-na? It's a what? Electron transport chain. And in a na-na-na-na-na, an electron transport chain, you create a hydrogen ion gradient. Where are those hydrogen ions going? Right there in the in the interior of the thylakoids, the hollowness. So that when they go back out, they will make some ATP. Out here is where the ATP will be made. It'll go out into the stroma, the space around the thylakoid membranes. Another thing your na-na-na makes is on photosystem 1, it makes some, what? Reduced NADP. So now you have the reduced NADP out here in the space around the pancakes, and you have ATP. Take some ATP and some reduced NADP to the or the light-independent reaction. Exactly, which is occurring out here around the outside of the green pancakes. And it's there that you will have your ATP, your reduced NADP, that you have to fix the CO2, reduce the CO2, so you can make some sugars and regenerate some RUBP. Okay, so here's an actual chloroplast, and you can see those stacks, the grana. And then here, it's inside of here that you have those hydrogen ion gradients, so when they go back out, they make ATP. And you reduce your NADP, and you do out here is where you're going to do your dark reaction, Calvin cycle. Okay, and you have that picture too to help you with that. So it starts with light of some sort. It doesn't matter if it comes from a flashlight or the sun. We have the electromagnetic spectrum. And we know that the smaller the wavelength, the more what it has? Energy. So who has more energy, Roy or Biv? Biv. Roy is lazy and big. Big wavelengths and lazy. And when we look at the electromagnetic spectrum, where visible light is. And so plants appear green because they are what? Reflecting green. Now, is green always reflected? No. There are other pigments, like what? Carotenoids. They absorb green and they reflect orange. So they work fine with the green wavelengths of light. We talked about the changing seasons. The changing seasons, right? And as winter approaches, the chlorophyll breaks down, and you have these other pigments, the oranges and the reds, that work with the larger wavelengths of light, right, as we go into fall. You want to know the equation for photosynthesis, right? So you can talk about how CO2 gets reduced into glucose, water gets oxidized into oxygen, and the hydrogens that are here temporarily go to NADP, and then they are given to CO2 in order to build that glucose. Okay? And we looked at this graphic. This light reaction right here is occurring on those green. Wow, did you see that? Just changed to blue. What color is it going to be now? Oh, okay. Interesting. Okay, and so we concentrate the hydrogen ions inside those thylakoid membranes, so when they go back out, this is when you can make the ATP that you need for the Calvin cycle. And when we we can give it to NADP, forming reduced NADP that we need to use in the Calvin cycle, right? And then from the Calvin cycle, we're cranking out sugars, not whole sugars. What do we crank out? Halves. What do we call them? PGAL. It's a P-gal. P-gal. PGAL and 3P. Good. That's fun. Okay, I'm going to continue. I'm going to ignore it. I don't know what I did. All right, now we looked at this picture as well, showing the reactants and the products for the light reaction, what you need in the dark reaction. These two electrons right here from NADPH, who did we get those from? Water, right? The hydrogens donated their electrons, and then you're just left with the oxygen, right? And then once those electrons are given to CO2, then the NADP goes back to oxidize, and it can be reused again and again. And once you've spent the ATP, it can go back and be reused in the light reaction again, so you can make more. All right, then we looked specifically at the light reaction that occurs. And we can see here at the bottom, most simply, that water is split and oxygen is released. The hydrogen's electrons are going to get what? Excited by the sun's energy. They're going to na-na-na-na-na-na. Then we know there's something there called what? PQ, who can help bring hydrogen ions to the inside of the thylakoid membrane. And we like that because the hydrogens come in, when they go back out, you can make some ATP. Same electron gets excited again by the sun, but we don't go all the way down in energy. We stay powerful and take NADP oxidized and make it what? Reduced. These same electrons are going to be given to CO2, and with energy, we can then make our halves of glucose. So the electrons that are in glucose ultimately came from what? Water, which was formed as a result of cellular respiration. Who catches at the bottom? Oxygen forming water. Okay. So let's do just the light reaction. Ready? Here we go. Water, 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 water. What are we going to do? Break it in half. What do we get? Oxygen and electrons. Sweep, sweep. What are we sweeping? Hydrogen ions that we took these electrons from, right? We sweep, sweep. We are in photosystem 2. It was discovered second. Okay? Sweep, sweep. For a second. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Na-na-na-na-na-na. When we're na-na-na, we're just moving along that thylakoid membrane, right? We're na-na-na-na-na, lowing in energy, right? Free energy. Okay, here we go. Na-na-na-na-na-na-na-na. PQ, bringing in more hydrogens, and you make some ATP. Now we are in photosystem 1, and it was discovered first. Here comes the sun, traveling in waves. Particles of light are called photons, and it gets the electrons excited. Na-na-na-na. Who catches it? Why are our hands up so high? We have a lot of energy. We're not low on the free energy scale. We are high. Okay? NADP forming reduced NADP. Then you take some ATP and some reduced NADP to the dark reaction, or the Calvin cycle, or the light-independent reaction. Good. Ask the person next to you if you have any questions on that. Are you golden pony boy? Okay. This is called non-cyclic photophosphorylation. Non-cyclic because electrons come in via water and go out via reduced NADP. Via means by way of. Non-cyclic. Photo, meaning you need light. Phosphorylation, we are phosphorylating ADP to make ATP. All right? There is such a thing as cyclic photophosphorylation, but all that gets us is ATP, no reduced NADP. It's used as backup for non-cyclic, just like we use right. Well, we don't use the lactic acid so much as we use anaerobic respiration for backup for aerobic. Yeah. All right. Now let's talk about the dark reaction, Calvin cycle, light independent reaction. So step 1 is going to be able to fix this CO2. Now CO2 is a C what? 1. CO2, 2 oxygens, 1 carbon, right? And we're going to fix it to a compound in here that is a C what? 5. What is that C5's name? RUBP, which we commonly call a gardening tool. Okay? So we have a C1 plus a C5 gives us a C what? 6. So now let's just do 3 at a time. So 3 C1s, dang it, plus 3 C5s gets us 3 C6s. But we know when you hook a gardening tool up with a 1, it makes a very unstable relationship, right? And all the C6s do what? Break in half. So instead of having 3 C6s, since they all broke in half, what do we have? 6 C3s. But now we're going to use our ATP. We're going to use our NADP reduced. And now we're going to have 6 high energy C3s called PGAL. G3P. And we could walk away with those 6 C3s. And we would have 3 glucoses in essence, right? But we're not gonna. What are we gonna do? Take away 1 C3, right? Take away a single PGAL G3P. That's all we're gonna take away. Then what are we gonna be left with? 6 minus 1 is 5 C3s. And then what are we gonna do? We're gonna invest some more ATP and rearrange these 5 C3s into 3 C5s. And do it all again. And if we do the whole thing all again, then we will have 2 of these, which is equal to 1 glucose. Okay, and we looked at that here. We looked at that here. We used these and moved them around, remember? And then we ended up being here. Okay, questions or concerns on that before we sing that part? Okay, here we go. 3. 3C5 plus 3C1 makes 3C6. Pause. 3C5s are referring to what? RUBPs. 3C1s are referring to what? Carbon dioxide. Okay? 3C6. Break down to 6C3. Use some ATP. Use some reduced NADP on your 6C3. Take 1 away, PGAL, G3P. Now, all 6 of them were PGALs and G3P, right? We just took 1 of those away. Okay. Okay. Take 1 away, PGAL, G3P. And you're left with 5C3, which are the PGALs, right? Use some ATP to build 3C5 plus 3C1 makes 3C6. Break down to 6C3. Use some ATP. Use some reduced NADP on your 6C3. Take 1 away, glucose. And you're left with 5C3. Use some ATP to build 3C5. Stop. Okay. Now, we know plants can take that G3P, and yes, they can build glucose if they put 2 of them together, but they could also build fructose or sucrose or fatty acids or amino acids or starch or cellulose. G3P is the jumping off point for everything that they need. And in fact, it saves them 2 ATP in what? Cellular respiration. Because the first step is to take glucose and break it in half. That costs you 2 ATP. They just start with it broken in half, and they're already ahead 2 ATP. Okay. And let's talk about naughty. Okay, RUBP, which is our 3C5. If CO2 runs low, it hooks up with oxygen, and that's called photorespiration. Not helpful. Not photosynthesis, not respiration. So when you look at most plants, they run the risk of that. Most plants are called C3 plants, and they run the risk. If oxygen levels are high and CO2 levels are low, they will do photorespiration in their mesophyll cells. And this is what normal plants look like. Here you can see these big green photosynthetic bundle sheath cells, and these are called C4 plants, and they have a compound called what? Yes, Pep, I was trying to be a cheerleader. Very peppy. He's like, I will help you, I will be faithful. I will only bind to carbon dioxide. Come on, carbon dioxide, let me drop you off at this bundle she cell. There you go. RUBP is not tempted by the fruit of another. And so as a result of that, yeah, you can't do as much dark reaction, can't do as much Calvin cycle, but the payoff is good because you don't have to run into as much photorespiration. That is a partition in space. We're saying we do dark reactions here. A partition in time, oh, this is your space, like corn, maize, okay? Partition in time are CAM plants. CAM plants fix their CO2 with their PEP at night when they can afford to have their stomata open. They're not worried about water loss because it's cool in the dark of the night, but they can't do any of the light reaction then. So it's like, okay, great, we have all this CO2, but they wait until the sun comes up, stomata closed, PEP releases CO2 to the interior, and then they can do their dark reaction then. And then we just kind of compared photosynthesis with cellular respiration. Got it? Good? You want to take a practice quiz? Okay. All right. Sorry. Study hard. Use your practice questions we did in class. Love you. Have a piece of toast if you're hungry. Don't stay up too late.